Hello, everyone, and thank you so much for joining us here today in the Garvan Auditorium. It is just wonderful to see so many of you here face to face, and a big hello as well to everyone who is joining us online, on Facebook Live and via Zoom. I believe we have around 300 people from across the country joining us, which is just wonderful. It's lovely to see so many familiar faces and also a few new ones as we meet here today, united by what is really a shared mission to better diagnose, prevent, treat, and ultimately cure the conditions that have the most profound impact on society. My name is Dr. Vivian Richter, and I am the Communications Manager here at the Garvan Institute, which means it is my great pleasure to work with the scientists in this incredible building to share their research with everyone, including you, our wonderful Garvan family. Now, if you recognise my voice, it might mean that you've listened to an episode or two of the Garvan podcast, Medical Minds, which I would highly recommend you check out. Now, before we begin, I would like to just start by acknowledging the traditional custodians of the land on which we meet, the Gadigal people of the Eora Nation, and also the traditional custodians of the land Australian Bioresources is on, the Gundungurra and Darawal people. We recognise their continuing connection to land, waters, and community, and pay our respects to elders, past, present, and emerging. Now, just to start off with a little bit of housekeeping, we will have 3 presentations today, followed by a Q&A. Now, I encourage both our audience online and in the room today to please raise your hands with questions. For our online audience, if you'd like to, at any point during the seminar, type in your question at the bottom of your screen in the chat box. We will get to them during the Q&A. And for everyone in the room, please just raise your hand and a member of the wonderful Garvan staff will come over with a microphone. Our bathrooms are located just outside the auditorium on the ground level, and if you haven't already done so, please make sure to switch off your mobile phone so we are not disturbed today. Thank you. Now, we are here today to talk about multiple sclerosis. Not everyone in this room today will know someone living with multiple sclerosis or MS, but this is a condition that affects more than 33,000 people in Australia alone and affects more young people than any other acquired neurological disease. The exact cause of MS is unknown, and there is currently no cure. And the reason that we're all in this room today is because we find hope in the groundbreaking research that will bring us closer to early detection, prevention, and ultimately a cure. Now, you will hear today from 3 researchers who are key members of the Open Coast to Coast Australian Multiple Sclerosis Consortium. It's a little bit of a mouthful. It is a world-class team of more than 40 clinicians and scientists who are rising to the challenge of investigating a new approach to understanding multiple sclerosis. We will also hear from Mr. Mark Alicia, who lives with multiple sclerosis and is a member of the OCCAMS Lived Experience Advisory Team. Mark, it is a real privilege to have you in the room with us today to share your perspective. Thank you so much for being here. Now, this brings me to introduce our first speaker today, who is the scientific lead of the OCCAMS Consortium, Professor Tri Phan. He is an immunologist and the co-director of the Precision Immunology program here at Garvan. He is an early adopter of cutting-edge microscopy and single-cell genomics technologies and a bit of a superstar here at the Institute. Please join me in welcoming Professor Tri Phan.
Thanks so much, Vivian, for that very kind introduction. And thanks everybody for coming and joining with us today, and also a warm welcome to people online. So it's my great privilege today to introduce to you something that I think has been bubbling under the surface for a few years, but it's really come to fruition in the last few years, which is the creation of this OCCAMS consortium. So what I'd like to do maybe in the next few minutes is introduce to you the problems that we want to solve, the people that we think are going to be critical in solving it, and give you a sense of how we're going to do that. So I guess I'm just going to begin by telling you that multiple sclerosis as a disease really, came into existence really in 1868 when a French neurologist and pathologist named Jean-Martin Charcot described that in these young women with these debilitating diseases, which were previously ascribed to hysteria, had these plaques of demyelination in their spinal cord and in their brain. And so the French term for it was sclerose en plaques, and so the term multiple sclerosis was coined back in 1868. And it's been more than 150 years since then, and along the way, I think a lot of discoveries have been made, and I think it's been quite gratifying, particularly in the last few years with some very amazing results in terms of new treatments being discovered, but still I think you agree that there's still a long way to go in terms of our understanding of multiple sclerosis. And really the most important thing, which is how do we prevent disability from arising in the first place? So the impetus for the OCCAMS consortium and a lot of the sort of inspiration for the research in the last couple of years have come from this seminal paper that came out a couple of years ago, which I think finally definitively really linked the development of multiple sclerosis to a virus called Epstein-Barr virus. Now, this is something that's been suspected for a long period of time, that maybe multiple sclerosis is something that may be triggered by a viral infection. What these investigators did, which is quite an extraordinary testament to the power of big data, I guess, is they tracked 10 million people over 20 years. And they were able to examine 60 million blood samples to show quite definitively that in people who develop multiple sclerosis, it was preceded by the acquisition of Epstein-Barr virus. And what this shows then really quite convincingly, I think, is that Epstein virus was necessary for the development of multiple sclerosis. But it wasn't sufficient because, as we know, Epstein-Barr virus is something that infects more than 90% of adults, and not everyone gets multiple sclerosis. So the big question now is, what's the missing link? What do you need in addition to infection with Epstein-Barr virus to then go on to develop multiple sclerosis? So what is EBV? Well, so EBV is a really common double-stranded DNA virus. You may know it as the kissing disease because it's commonly transmitted in adolescents in their saliva when they kiss, and in a third of individuals, they're symptomatic and they develop glandular fever. And EBV also can cause quite severe diseases like Burkitt's lymphoma, and particularly in people who are heavily immunosuppressed with drugs or people who have HIV or AIDS, it can cause lymphoma. And that's because EBV can infect not only the epithelial cells, but also the B cells. And B cells are a really important cell in the immune system. So typically we would imagine that EBV would, be acquired from the saliva, and in the tonsils, there are epithelial cells that can get infected. And so in the epithelial cells, EBV sets up what's called a lytic infections. So it infects the cells, replicates, and then bursts the cell open to get out and infect other cells. But of course, it's not just the epithelial cells in the tonsils that get infected, because it can also infect the B cells. And here it does something much more insidious. And what it does is it hides within the B cell and enters what's called a latent phase. So people can be infected and then have EBV inside their B cells for long periods of time after they're infected. And it's that latency that allows the development of these other diseases. So one of the questions that we really wanted to understand then, what is it about the virus and what is it about the immune response to the virus in individuals who go on to develop multiple sclerosis? And so as you can imagine, the whole point of this slide is just to tell you that this is actually a really complex problem. And the life cycle I just described is a very simplified version of what's going on. And so usually in immunology, when we show you really complicated slides like this, is really another way of saying we really don't know what's going on. Well, I mean, it's true, right? I mean, we have a lot of theories, and as immunologists, we're really good at coming up with theories. But I think the real challenge now is actually to test those theories and then come up with important discoveries that actually might help guide us in understanding, what ultimately is the goal, which is to find out what is it about the immune response in susceptible individuals that make them go on to develop EBV, and how would we get to the bottom of that? So OCCAMS is OCCAMS razor, you may have heard, is a principle in not just in medicine but in a lot of fields, and it's attributed to this Franciscan friar who in the Middle Ages came up with this concept of the law of parsimony, which is the idea that entities must not be multiplied beyond necessity. And then it's sort of like paraphrased as the simplest explanation is usually the best one. So what we realized is because we're dealing with a complex problem, it was going to be really difficult to solve on our own. So what we did was we set up this consortium, which conveniently, and as you know in medicine we love our acronyms, conveniently shortens to OCCAMS. And so this is the Open Coast to Coast Australian Multiple Sclerosis Consortium. And while OCCAMS razor was all about simplifying things, what we realized, okay, so those of you who like bad puns, you can also split it up and call it the OCCAMS because, we're Australian. So instead of simplifying things, what we realized was that actually, this is a complex problem. And so OCCAMS was about, not multiplying beyond necessity, but here by necessity, we're actually going to need, a multidisciplinary team. It's not going to be solved just by neurologists seeing the people with MS. It's not just going to be solved by the clinicians and the basic scientists working in isolation. What we really need to do was we needed to bring all these people together, the neurologists, the immunologists, experts in autoimmunity, MS, virologists who truly understand EBV, and also people who understand genomics and gene-environment interactions. Because at the end of the day, what we need is really different, diverse ways of thinking to bring to the problem. And really importantly as well, we needed to interface with the people living with MS. And so we've been very lucky to have people such as Mark on board, and also the support of MS Australia. So we're really grateful for that. So what we did was we brought together, as Vivian mentioned, a very big group of Australian experts. And we're a plurality. We are not one concept or one idea. And I think one of the secrets, for our success moving forward will be our ability to, take on diverse ideas and opinions and test them rigorously, and then, work through them to get to a solution. So we have, as you can see here, involvement not just in Sydney, but across Australia, in Melbourne, in Perth, and in Tasmania. And also, it's not just Australian experts. We've also gone internationally. We have experts in EBV, in autoimmunity, in MS from across the world as well. And so, who are the people involved in OCCAMS? You're going to hear from two of them today. Dr. Jennifer Massey, who's the clinical lead, and truly, is the inspiration behind the whole endeavour, and Dr. Seyhan Yazar, who's here with us today, who's driving a lot of this discovery science. But as you can see, there are a lot of people on this slide, and that's kind of like, that's not just our family. We have a much larger family. There are many more people involved than that. And then we also have an extended family as well, of people who are going to be really critical for our success, because OCCAMS, while we're funded initially for a few years to get the research off the ground, the key thing for us moving forward is to have a sustainable program, because this is a complex problem that's going to take many years to solve, and it's got lots of moving parts. So everyone's role is going to be critical in this. So our mission is really quite simple. We want to stop MS. At the moment, there are many effective treatments, but the problem is that once the damage has been done by the demyelination, at the moment anyway, we cannot reverse that. And the other problem, of course, is that not every individual with MS will respond to the treatment. And sometimes you will have this sort of like cascading treatment where you try one and it doesn't work and you try another. And so in the precision immunology program at the Garvan Institute, what we're really keen on is understanding this heterogeneity, why people are so different in terms of their responses to treatment, why they're so different in terms of their disease journeys, and how we can work out and predict what those differences are so that we can tailor individuals to the treatments that's going to work best for them. What we want to do is to shorten that period between, you know, the inflammation, the damage to the central nervous system, and the beginnings of the institution of effective treatment. So what we really want to do then is a way to predict who will get MS and then be able to prevent it before it happens. And really to stop MS, ultimately really what we want to do is to be able to develop a primary prevention strategy that would actually, you know, in susceptible individuals such as first degree relatives who may have, you know, a biomarker that puts them at risk, to be able to, you know, give them that treatment and then stop them from going on to develop disease. I'm going to very briefly touch on the 3 arms of the study that's involved, and I think Dr. Massey and Dr. Yazar will go into details a little bit more. But essentially the first thing we want to do is understand why is it that different individuals have such different responses to the virus, and why and how those responses set them up to then go on to develop MS as an autoimmune disease. So this involves a lot of functional studies, deep immunophenotyping, a lot of genomic studies, and then, you know, being able to then integrate all that knowledge is going to be really critical. The second thing we're going to do, which is, you know, really taps into the unique talents and abilities on campus here as well, is we're going to track the T and B cells that are damaging the immune system and that might be reactive against EBV across these different immune compartments. So the damage is occurring in the central nervous system, in the brain. So we're going to collect cells from the cerebrospinal fluid, but also the immune response is staged or is organized in the draining lymph nodes. So we're going to get cells from the deep cervical lymph nodes. And then we're going to also look at the cells as they traffic through the blood. So being able to track cells through these different compartments, through their very unique clonal receptors that allow us to also determine what they're reacting to, I think will be really critical in understanding how these responses are different from one individual to the next. And then finally, I think what's really speaks to the strength of the whole OCCAMS consortium is that what we're going to do is bring all this together. So we have all these different expertise, and what we're going to do is then integrate all the data that we generate and use machine learning approaches to then develop a predictive biomarker that takes into account not only the results of some of these studies, but also the things that are, you know, really important as well, which are the clinical aspects, right? Being able to integrate both, you know, these different modes of information. And the endpoint then, hopefully then, is to end up with a predictive biomarker that's going to tell us who's going to get MS. And hopefully then we'll be one step closer to our ambition, which is to stop MS. Okay, so I think that's my cue then to hand over to my brilliant friend, Dr. Jennifer Massey, and the clinical lead for OCCAMS, and also the person who really is the inspiration for the whole endeavour. So, Jen.
Thanks, Tree, and thanks to everyone for joining us today, both in person and online. It's a real privilege to be able to talk about this research, and we've been incredibly well supported by the Institute, so thank you to Garvan. So I'm a neurologist, I'm a clinician. My understanding of words like big data and, well, that I kind of understand, but single cell sequencing, all of this is beyond me. So I'm going to take you a little bit from I suppose, a clinical perspective about what are the problems that we face in MS and why these problems are so important to try and solve. So Tree has already introduced us to this gentleman, Charcot, who coined the term multiple sclerosis or sclerosis on plaque. And you can see the picture here, which was his drawings about plaques within the spinal cord. And we can now, it's actually just over 50 years since the first MRI scans were able to very clearly identify these plaques in vivo. So that's really important when you're considering a trying to diagnose a condition that affects the brain and spine, not areas that we can easily take tissue samples from. So we're reliant very heavily on MRI scans to help with the diagnosis of MS. But actually about 500 years before Charcot first described this condition, it was already being reported in literature of the time. So this was a Dutch girl, Ludwina. She was actually the patron saint of ice skating. And at the age of 16, she was characterised to have developed this relapsing illness whereby she would lose power and coordination. Years later, she became blind in one eye, developed weakness and pain, and those are the symptoms that still plague many people with multiple sclerosis. So multiple sclerosis is defined as a demyelinating condition. What does that mean? So our nerves within our brain and spine are coated with myelin, and that's kind of like the insulation that enables messages to get communicated instantly. You know, if you want to pick up or move the mouse, I don't have to do that, over a seconds or minutes period. That happens instantly when I ask my hand to move the mouse. And with demyelination, loss of that myelin, there's slowing of messages getting from the brain to the limbs. Sorry about that. We also know that the nerves themselves, when they lose their myelin, they don't do well. And so the problem is not just loss of myelin, but also, unfortunately, nerve damage, which is quite frequently irreversible. So multiple sclerosis is a lifelong condition, and as Tree mentioned, currently we do not have a cure, and that's what we're working towards. It's a condition that is typically defined by these relapsing episodes where someone develops symptoms that may last days, weeks, and those symptoms typically abate, and it might be 2 months, 6 months, 2 years until another relapse occurs. But over time, as you can see, our brain and spinal cord's ability to deal with those relapses is limited. And over time, we see what's termed progression, which is when the neurological system basically fails to compensate with these multiple attacks. And unfortunately, disability continues despite our best efforts with therapy. Interestingly, we now know that MS probably begins many, many years, perhaps even decades before the first defined relapse. So some very nice work out of centres in Canada and the UK have shown that even in the 5 or 10 years before someone's diagnosed, they're attending GP, they're attending emergency department more often with symptoms that many of us would consider to be sort of silent: headache, migraine, pain, itch, and other symptoms that are very hard to pinpoint as being related to MS. And so we can see this really nice period of time where if we were able to diagnose people earlier and intervene, we could prevent the irreversible neurological damage that happens later in the condition. As Tree mentioned, we don't understand completely what causes MS, but we are aware of genetic and environmental factors. But this is not an easy space to understand, certainly not for a clinician like myself. So we know there is one gene that is more common, about twice as common in people with MS than it is in the general population. So you can see the dash bars here are people with MS. But that's still only sort of present in somewhere between 30 to 50% of people with MS, and it's present in 20 to 40% of people who don't have MS. So it's a little piece of the puzzle, but it doesn't tell us much more than the fact that this condition is, this gene is increased in some people. Big genetic studies have tried to define exactly what mutations in other immune genes might be relevant, and we've found lots of little hints but no big answers. But we know that this condition is driven by the immune system, and particularly, as Tree mentioned, certain cells in the immune system known as the T and B cells. Environmental factors have long been of interest in MS, And the biggest proposed culprit has been this Epstein-Barr virus. So even before that big study with 10 million individuals, we've known that it's about 3-4-fold more likely to develop MS if you've been exposed to this EBV virus than without. And this is the same paper that you've already seen, showing that in all of these individuals here, which are the horizontal lines, they developed EBV. The blue bar began before they developed MS, which is the orange dot. So to summarise what we do know about MS, we know that these relapses are caused by immune cells, here purple cells, coming out of the blood vessels into the brain and attacking the myelin, which here is this sort of orangey yellow coating. I mentioned that it's really hard to study MS because of the very nature of the tissue that it affects, but we have had unfortunate cases where people have died young with the disease, or cases where we've managed to take some brain tissue, and we can see these cells here, the little speckledy dots, leaving the blood vessels and travelling into the brain. And we know that this is a complex interaction whereby those cells travel via the lymph nodes in the neck. And that's why we're interested in testing the blood and the lymph tissue of people with MS to try and get hints at what those cells might be reacting to. Because as Tree mentioned, there's a missing factor that we don't fully understand. I spend most of my time seeing patients, and I wanted to take you through the journey of a patient of mine, just to highlight the importance of these questions that we're trying to unpick. So Mrs. SF is a lady who I first met in 2017. She's originally from the UK, but she's been living in Sydney for some time. She came in with dizziness and what we term in neurology oscillopsia, which is this sense of movement even if your eyes are looking still. And on examination, there was a problem with the coordination of her eyes. So for you to see one image, your eyes have to move synchronously. And when that doesn't work, people get blurred or distorted vision, and it's called internuclear ophthalmoplegia. And it's a very common sign of an MS attack. She had an MRI scan of her brain, and in essence, these white areas of inflammation here that you can see across her scan were very typical of inflammation caused by MS. And even within a few weeks of her being diagnosed and starting on treatment, she unfortunately developed new symptoms, numbness of the face and unsteadiness with walking, and had gone on to have more spots on her scan. So this is the type of situation that keeps me up at night. However, we have made huge advances in the treatment of MS, and she started on a drug called natalizumab. It's basically a drug that blocks white blood cells from getting into the brain and spine, and she's been on treatment here and has done incredibly well. So we've made big steps, but there's still a long way to go. You can imagine if that you don't have an immune system controlling or protecting your brain and spine, that you're at risk of infection, and that's a serious issue for all of our patients. She's stable at the moment, but this condition remains incurable, and that's a lot to deal with for someone that's diagnosed in their 20s. Most of our patients are diagnosed between the age of 20 and 30. And unfortunately, what's damaged cannot yet be reversed. And so this disability that we aim to prevent, in this case, is going to be a permanent issue for Mrs. SF. The other big question I hear from many patients is, what does this mean for my children? Is this a condition that I'm going to pass on to my kids? And so our goals with the OCCAMS consortium is to use blood, lymph tissue to try and find out what's driving this condition. If we can identify people in that early prodromal period, then that's an opportunity to intervene. And ideally, we're going to develop risk scores that will enable us to screen family members of people with MS before they even come to be diagnosed. And to explain how that's all going to work in 10 short minutes, I'd like to invite my friend and colleague, Dr. Seyhan Yazar, who's the group leader in the Precision Immunology Lab at the Garvan, to explain the science. And she's the person who's going to put all of this complexity together. So thank you.
Thank you, Jen. And hello, everyone. And it's a pleasure to speak here and to tell you about our great research that, trying to stop MS completely. So my role is a scientist, a computational biologist, as someone who has experience in population genetics, is to marry the basic science with clinics. And that's why, I think it's a very great opportunity to work in the OCCAMS consortium. So throughout my talk in the next 10 minutes, I'm going to tell you a bit of details of the research approach that we are taking to connect the connection, to examine the connection between EBV and multiple sclerosis, and how we are going to actually translate our findings into clinical practice. So as you have heard from both Jen and Tri, we have 3 different study protocols. We have established, but in the interest of time, I'm going to tell you about the first one. And actually, this protocol itself has some sub studies. So I will just talk about one specific one. So you heard from Jen and Tri again that in Australia, EBV infects more than 90% of healthy individuals, and yet MS only occurs in less than 1% of in 1,000 individuals. So what makes some of us who are infected to the EBV to develop MS? So this is a question that we are trying to understand, and we think that one of the answers lies around our genetics, and this is one of the primary questions that we are trying to untangle in the OCCAMS consortium. So I will briefly tell you about what we know so far about the genetics of MS. So if you have a family history, if you have a family member, especially in the first-degree relative, you have actually 1 in 7 chance of having MS, develop MS, and this increases in identical twins much greater, up to 25 to 30%. When we look at the studies looking at what is the genetic contribution of genes and environment, we see that 50 to 70% of MS is actually caused by genetics. And so this estimate is comes from estimations of looking at both twin studies as well as population studies. So what is that so this 50% of genes, so what is it made of? We are actually trying to identify these by doing large cohort studies. As we see an example here on the slide that what we call a genome-wide association study, where they have actually compared DNA from 50,000 multiple sclerosis individuals and equivalent controls. And we have only identified more than 200 variants or several a few genes that were identified associated with MS. And when we looked at these genes that have been associated with associated with MS, they are actually involved immune system, and hence there is a high chance that immune system is responsible for the beginning of definitely the beginning of multiple sclerosis development. And when we look at the immune system, it is actually a very complex network, which includes more than 1.8 trillion immune cells, and which each cell actually have its own sets of expressed genes, and the complexity increases within each cells because there are millions of different molecules and molecular interactions, so it adds to the complexity. But underlying that complexity is something that very basics that manages it. And this is something that we probably learned very early in our lifetime. If you remember from your high school years, the concept that DNA makes RNA and RNA makes the protein. So our DNA, the 4 letters that we have, is actually managing what is happening in our body. So just to give you an example, so we have the letter T here, in our DNA in a certain position, which helps us to have this RNA molecule. So if I change this T letter into an A, does it going to allow me to have this this RNA molecule, or is it going to reduce the amount of that I'm going to get at the end? And so this is the basic question that we are trying to understand in the OCCAMS consortium. And so how we are going to study this detailed detail in the OCCAMS consortium. And so because as you heard from Tree and Jen, we are collecting samples from multiple individuals, and each individual has a lot of cells. So how this is going to happen? And so I'm going to take a step back and tell you about what happens once we receive the samples from Jen and other clinicians when they come to the lab. So we don't process all the samples at this once they arrive to the lab. We actually store them in liquid nitrogen, because if we process them and treat them, then we can create technical errors that can actually impact our data analysis. So we store all of them at the moment, and once we finish collecting our samples, we will tell these samples and then prepare them for centrifugation, a step to actually separate the blood into different layers. And centrifugation is a method where you are using the gravity to actually separate, to make that separation happen. So once we have separated the blood, we have these layers. The immune cells are the ones that we want to take from here, which are also known as peripheral blood mononuclear cells. And they are called peripheral blood mononuclear cells because they are circulating throughout body, and they have a round nucleus. So I'm not going to explain to you the rest of the sequencing process, but I want to tell you a bit of the history of sequencing and why it is important to do this research now and here. So there have been traditional technologies to measure RNA, and it has been in the scene of scientific discovery since mid-90s. So the first method, which is known as quantitative polymerase chain reaction, is actually discovered in 1980, although the Nobel Prize was given in 1993. And this is the time that when this technology took off. So in this technology, in briefly, what happens is we are generating specific markers to actually measure the target genes. So it is a bit cumbersome, and we can only do a limited number of genes per experiment. Then the macroarray technology came in, and in this one, I can give you a bit of analogy of if you have a chessboard with millions squares, and each square is representative of the gene. That's the way that we have actually had the macroarray technology. So we were actually putting the blood sample into this chessboard, and then the genes that are what's present in the blood were actually matching to the squares that the genes were expressed. So, and that's how the macroarray technology works. And then, of course, in the later on, we have actually had with the advancement of high throughput sequencing, which means that now you can actually generate data for not only tens of people or hundreds of people, but for thousands of people and more. And that's the bulk RNA sequencing. But the real interesting and I guess the exciting technology was the single cell sequencing, which came into play in 2009. And so I'll just give you the difference between bulk RNA sequencing and single cell RNA sequencing, why this is the right methodology to do this research. So if you think, let's consider a fruit shake. And if a fruit shake, it is homogeneous. We won't be able to know the fruits that we have there by just looking at it. And this is akin to doing bulk RNA sequencing. But in the single cell sequencing, we can actually separate those different fruit layers, and we can identify which ones we have in the fruit shake. And this is very similar to like, in the single cell sequencing. And so when we can only like, look at the gene expression in a population in bulk RNA sequencing, in the single cell sequencing, we can actually separate the cells in each cell and look at expression of like different genes in each cell. And this is like, very important to understand the cellular differences to understand like in the EBV related MS signatures, and especially specific cell types like B cells that like both Jen and like Trey mentioned. But so the emerge of like single cell technologies started in 2009, but that's not where it ended. It is like continuously evolving, and there are new technologies and coming on board. And Garvan's genomic platform here houses many of these like technologies. So we are very lucky to actually have these like technologies at home to actually do our research. And so looking at like, all the possibilities like, that's available in the technology, we have decided to do something different. And I will explain to you why we choose this method in a bit. But we have chose to go with like, multi-omic sequencing in this like, in our study. And so what is single cell sequencing and multi-omic sequencing? In addition to RNA molecule extraction from a cell, single cell multi-omic sequencing allow us to get another type of data, which is called chromatin accessibility, which is, you may have heard the term like, epigenetics in nowadays, it's very popular, which is like, is another important piece of information impacting our like, genetics. But if I give you like analogy to make this like, a bit more concrete in your mind, think of your body as a city of houses, and each house is a cell within your body. And so in the cell, like in the house, you have lots of rooms. So each room is like the gene that you're expressing. And so, and to go into a room, you need a door. So the chromatin accessibility is actually the doors that are present, like, to go into a room. So it is therefore, it is very important to have this, like, additional piece of information. And so the other reason that we are interested in epigenetics and chromatin accessibility is Epstein Barr virus is like, known the master and manipulator of like epigenetic mechanisms, especially in cancer. And so, and that like actually it is related to like one of the famous hypotheses in cancer research is like what happens is like EBV virus infects a cell, it goes and like change the DNA and make changes in the epigenetic programming, and then it just like leaves the cell. Then we can't find any traces of the like, the virus itself, but when we want to look at like, the next generation cells coming up from this original cell, the epigenetic changes that has left behind is actually remain. So, and that's perhaps like, one of the ways that like, the virus is impacting the cells and like, and leaving like this programming to continue and involve in disease development. So going back to like, our aim of like identifying cell type specific MS signatures is how we are going to do it, and does it really exist? So what is our basis for such research? And so, and we know this actually exists because I was involved in a large study looking at multiple sclerosis signatures in healthy individuals. So these individuals did not have like multiple sclerosis. They just like had the susceptibility, like, genetic code. And so when we looked at like, this large data set, what we have seen, as you can see here, while some of like, genes that were shared among different cell types that is available in immune system, some were actually specific to like, certain cell types, as this gene here, EIF2, was specific to B cells, which is like, very important, as we have mentioned, like in multiple sclerosis. So we know these are present in healthy individuals. So this is why very important for us now to study the samples from who have like, MS and also like, people who have like EBV infection, but without like, of multiple sclerosis. So once we have identified these markers that are specific to cell type, then we can actually combine it with other large data sets, I have mentioned genome-wide association studies, and then pinpoint to causal genes that are involved in development of multiple sclerosis. And these causal genes then can be utilized and further investigated for therapeutic reasons. But another aspect that we have mentioned previously, that these findings that we will find from protocol 1 in this section will be really important for the study protocol that we have in study protocol 3, when we are actually trying to come up with a score to identify individuals who are at the risk of multiple sclerosis in population. So we are not stopping there. I would like to conclude my talk by mentioning you that we are already planning our next project focusing on MS prodrome, as Jen has mentioned to you. There are a lot to be accomplished, but we are very keen to find answers to these questions to stop MS completely. And with that, I would like to thank all our participants for their time and their support, but also I would like to acknowledge our investigators in this team making this research possible, and also our funding bodies. With that, I would like to invite both Jen and Trey and Mark-Alicia to this stage to join me for the panel, and our host, Vivian, to lead this. Thank you.
Thank you so much to all our speakers. Just bear with us a moment while we get set up for the Q&A. Have your questions ready. Hello. Wonderful. Thank you so much for those insightful presentations. I think we can all agree that that was a fascinating insight into what is really a very complex research question. Now, we've heard the science. I'd love to start the first question directed at Mark. Could you give us a bit of a personal insight into living with MS?
Yeah, thanks, Vivian. Living with MS, it's a real shock to the system when you're first diagnosed. As Jen described, most people discover they have MS in their 20s, and it tends to force you to make changes to your life that you never really expected to make, and it introduces a large amount of uncertainty into your decision making. As again, what Jen mentioned, you start asking questions like, how's this going to affect my kids? How's it going to affect my work? How's it going to affect, for me, it affected where I lived. I used to live overseas and worked overseas in a different field. Obviously, it's caused a lot of physical damage to my body. Yeah, those are the main effects, I suppose, of MS. It's uncertainty, it's damage to your body, it's damage to your mental health as well. But I guess on the other side of things, MS forces you to be very what's the word? You're reactive, and you learn to deal with changes quite well. I think people with MS are able to sort of think on their feet and come up with new strategies to deal with problems that most people would never really imagine. So there's good and bad. Yeah.
Thank you. You've been very closely integrated as the lived experience advisor to the OCCAMS consortium. Can you give us a little bit of an insight in what that collaboration entails?
Yeah, that's been a great experience. I've been a advisor with OCCAMS since last year, and with a group called Our Health in Our Hands, which works through the ANU since 2017. And it's been great because we're able to provide a little bit of direction into which way research might go. We're able to ask questions that clinicians and researchers might not necessarily consider. Even sort of some of the symptoms that people with MS experience that don't come up when we're face to face with our neurologists. We can bring them up, and you can find sort of common ground amongst MS patients that you might otherwise not expect. So for me, it's been rewarding. It's a way to take some control back from MS, I guess, and contribute towards finding a solution. Because as, again, what Jen mentioned, for me, I had damage to my nerves that happened very early on, and the treatments that were available to me prevented new plaques that we've described, but I've sort of, we haven't been able to reverse that damage. And if we were able to actually, prevent any damage from occurring in the first place, which is what this project is planning to try to do, that would be a superb outcome.
Thank you. You mentioned the guidance on the research. What would you say is the most pressing aspect? What would you love to see research address in the first instance?
The root cause would be great. I think that's the ultimate solution to this problem, because reversing MS is impossible at the moment. So yeah, figuring out what causes people to be more prone to developing MS, and then if we can figure out a prevention, that would be the ultimate goal, I think.
Thank you. I might now throw to the researchers to reflect on what this collaboration has meant on the research side. Maybe Tree, you'd like to take this one?
Yeah, sure. I mean, I think it's been really rewarding, because particularly as researchers, we're sort of like deep in the weeds, so to speak, staring down microscopes and looking at DNA sequences and everything. So, it's really important to have the input from people such as Mark. The things that are important to them really becomes the thing that are important to us, because ultimately, as we move forward, what we want to do is solve problems for people living with MS, not solve, esoteric conundrums that might only interest a few, professors somewhere in some obscure part of the world. So I think it really keeps it real. I think the other great value of it, though, is that that experience really informs for us, and I think where the best example of this is the direction that, and maybe Sehan can speak to this, that Sehan's going, which is trying to understand the MS prodrome, because that's something that we would not know if that much about without, speaking to people with MS and understanding, their experiences, and the fact that, for some of them, it's been quite frustrating that, they have these symptoms, they keep seeing doctors, but no one's been able to put their finger on it. But actually what these symptoms are telling us is that actually before they have the clinical diagnosis of MS, there's sort of ongoing damage and the immune system has gone awry. And that's a clue for us to actually, we need to look deeper into those sorts of situations as well. So it's been very informative.
Thank you. Do we have any questions from the audience? Yep, there's one up here.
Just in regards to the prodrome, the symptoms and signs that you had up, they're pretty non-specific, and they would apply to many conditions and diseases. So can you elaborate on that?
Yeah, that's spot on, and that's the challenge. So many autoimmune conditions, many non-autoimmune neurological conditions present with those very common symptoms. And many people have those symptoms without having any underlying diagnosis. I mean, put your hand up if you've had a headache, right? So the issue is trying to, I suppose, look at that subset of the population who have increased health usage and then take the information that we have from people who we do know have MS. And if we can identify a biomarker, for example, in the blood of someone who does have MS, look for that biomarker in people who are presenting with these more non-specific symptoms at an increased rate than the background population. That's the type of, I suppose, cohort or population where we would like to, for example, look at doing early MRI screening to try and identify people before they have any symptoms, or if we have a robust enough biomarker, make a diagnosis of prodromal MS or pre-MS and look to initiate treatment or prevention. And so the first part of our work is going to be focusing on identifying, I suppose, the signature in the immune system of MS, and then trying to apply that to, you know, the prodromal population or even relatives of people with MS, where we think there's an increased chance of being able to make an early or even pre-diagnosis.
Jen, I might stick with you for this one. We have a question from our online audience, which is around, can we explain why significantly more women are affected by MS?
Yeah, so this is an interesting feature that we see pretty much across the board in autoimmune conditions. And it's an issue that has been, you know, historically relatively underexplored. In a very basic sense, it's typically, you know, women of what we refer to as, you know, childbearing age, so people who have the hormonal profile of ovulating. And there is obviously an interaction between the hormonal profile and the immune response. And Sehan and another one of our colleagues in the OCCAMS consortium have done a lot of work on trying to unpick that. So I might even pass the microphone and the question on to Sehan to see if she could give a sort of brief explanation about that, but it's a fascinating area across autoimmunity.
Thank you, Jen. Yes, so from a genetic perspective, when we look at this discrepancy, in female versus males, it is across many autoimmune diseases. And Jen, said, this is an understudied area, but it is more becoming visible now. And so when we look at genetic causes, if you know that we have 23 chromosomes and going over we have 22 that are functioning autosomally, and we have the sex chromosomes. And so when we look at those sex chromosomes, they are not really well studied. It's because of the technological difficulties we had in the past, and now we are trying to overcome these. But we can still cannot find these cannot relate to these changes to our genetics. So it's not something that's happening in our DNA perhaps, but it's more the hormonal changes that we have in our body. Again, one of our, I haven't mentioned, but one of our focus in the OCCAMS consortium is to look at these sex-specific differences in the multiple sclerosis with my colleague from Dr. Sarah Baluz from UNSW. So we are hoping to give you a better understanding in the coming years for this.
Sehan, you had that slide, which the timeline of all the different technologies emerging. It seems this space is obviously incredibly active. Can you tell us why now is the right time to be embarking on this research?
Yes, so I can say that every week there's a new technology comes into play, because as I have mentioned, immune system is very complex. There are lots of different cell types. within those like different molecules in the machine. So how these like interact with each other, what is like, the outside, like, has an impact, including your like, what do we eat, how much like, sports we do. We are all like, investigating these like, in details. And to study these, you need like, very specific like, technologies. And it is now is actually these technologies are like developing more quickly with the advancement like, in other areas of like research, not only like, in science, but like, engineering, physics, and like, I think more recently is like, artificial intelligence. You will like know that like Nobel Prize was given to the machine learning program, which is like enables us like, to identify proteins better. So I think that's why like, now it is the time to like tackle these questions and to know that like, it won't happen like, we won't find the answers tomorrow, but we are like, much closer than before.
Thank you. Any other questions from the auditorium?
Hi there, probably a little bit of a follow-up actually to what was just mentioned. Could you just expand on the effect that AI will have on advancement in this area?
Yes, it is very exciting times for science, I think, because artificial intelligence can help us a lot, and I guess it will also make things quicker, things like we couldn't do earlier on. Definitely, like, so the process that I have explained to you, we're going to find these markers, we need to do a lot of like computational data analysis. And to do this, like, using our own, like, techniques that we have, like, statistical techniques, is like, is still doable, but it takes a lot of time. By using AI, we can actually accelerate those like, timeline. We can do analysis much more quickly. And the AI can help us like, to do things in more detail, and which is I think will be really integral to like doing research. On the other hand, I just wanted to mention that like, MS prodrome, and we have like, mentioned that like these like, symptoms are very common in the population, even like those who do not have MS, and that's very true as we heard. And so one way to actually that we are tackling that question is by applying AI techniques to electronic medical records to go through that like the unstructured data. So the doctor's notes that like, where we have like, it says that this patient had a fracture after like, taking this medication. So one thing that I think is really important when we are looking at this MS prodrome and where AI is going to be like, helpful is to understand us, give us the information about the temporal relationships, which I mean, when something happens, did it precede another event, or how many times it actually happened before? So yes, like, everyone has like a headaches, but I'm more certain that like, someone who is like, had a more in the inflammatory stage of MS is more likely to have like, multiple like, falls prior to their diagnosis. So we need to like, count the numbers. We need to count like, we need to investigate the events and the relationships, like, how one thing happens to the other. And AI is going to be key to answer such questions. Thank you.
Thank you. We've got another question over here.
Thanks, Vivienne. Hi, everyone. Fiona Mackay from MS Australia. This is more a comment than a question. I just wanted to say thank you so much to the team for doing this research. MS Australia has actually been involved for several years now with advocacy to the federal government for funding for EBV research. And I guess it's important for this audience to know that the federal government's absolutely backed in the last couple of years research into EBV and MS, indicating how important it is. So there were, a couple of years ago, there were $10 million worth of grants awarded to study this area. And we're delighted that this team of such a high caliber is progressing this research. I think it's so important, and it's really going to open opportunities for new strategies in prevention and in treatment of MS, and we're really excited to partner on this, so thank you.
Thank you. Might just go to one question in the Zoom room first. Is OCCAMS working on an EBV vaccine? I guess this is taking us to the environmental factors.
Yeah, sure. So there are actually multiple pharmaceutical companies that have developed EBV vaccines, including mRNA-based vaccines. So, for example, Pfizer's got one, Sanofi's got one. A lot of them have been developed as therapeutic vaccines, particularly to treat some of those more severe EBV-related conditions that I mentioned. And I think there's really an opportunity now to think about, EBV as a preventive vaccine, particularly in this context, because if all those individuals in that big study had EBV as the trigger that then revealed their predisposition to develop MS, you would imagine that if we could stop them getting EBV in the first place, we would be able to stop MS. So that's something that we're sort of exploring, hopefully, with some of these companies down the track.
Thank you. Now we are nearly out of time, but I would just like to finally throw to everyone on the Q&A panel about what your hopes are for the OCCAMS consortium.
My hopes that we, find out as much as we can, I suppose, and try to just better understand the immune system, how it's affected by EBV. I go into this sort of research quite open-minded, to be honest. Yeah.
Thank you.
So I guess part of the inspiration for our approach has been that, and I think kind of like alluded to by the question, is that there's another autoimmune disease, type 1 diabetes, which has a strong genetic component. More recently, it was shown that if you were first-degree relative and you had a biomarker, you had a very high risk of developing type 1 diabetes. What these people were able to do was that if you gave them an anti-CD3 monoclonal antibody, just one infusion, you could delay the onset of type 1 diabetes. And that was not one infusion. So what that does is it opens the door to the idea that actually it's not inevitable. If you can work out who's going to develop the disease, you can change their trajectory. And so my dream would be that, we would reach a time where there will be no new people diagnosed with MS because we've been able to stop it.
Thank you.
Yeah, I think I'd echo comments Mark made earlier. I mean, the biggest question that I see day in, day out in clinic is, what causes MS? And so it would be incredibly satisfying, I think, for people living with MS, but also as a clinician to be able to answer that question rather than that incredibly unsatisfying, we don't fully understand, or lots of, that's, I suppose, part of it. The second issue is, as Tree said, imagine being able to stall off this condition that, has plagued so many Australians and causes such significant loss of function in young people. That would be an incredible achievement.
Thank you. Seyhan?
So, as someone who is in the range of having the risk of multiple sclerosis, and also someone who's a mom of a toddler, so I think I am very interested in the question that Jen has raised, am I going to pass this to my children? So I guess my hope will be to say to a mom that no, you don't have to worry about this. We have a way to stop this, and so you will not pass into this devastating disease to your child.
Thank you. And please, could we thank one more time all our speakers today? Just acknowledging that there are many questions that came through on Zoom. Thank you so much for submitting those. Unfortunately, we didn't get time to get to all of them today, but please, if you have any follow-up questions, please feel free to email foundation@garvan.org.au. Just again, thank you so much for joining us today. We couldn't do any of this amazing work that we do without your support. And we'd just like to thank one last time our speakers today.